Welcome to the COVID-19 Researcher Spotlight Series. Today, I'm joined by Naama Geva-Zatorsky of Technion, the Israel Institute of Technology. Naama's group developed a protocol for detection of SARS-CoV-2 RNA directly from nose and throat swabs, without the requirement for RNA purification. Her group has also applied this protocol to saliva samples. The method utilizes reverse transcription loop-mediated isothermal amplification, or LAMP, offering rapid results that Naama and her team hope will enable successful community surveillance. Hi, Naama. Thanks so much for joining us today.
Sure, thank you.
I was wondering if you could tell our listeners a little bit about your latest COVID-related research.
Yes, happy to. We performed a collaborative research at the Technion together with the Rambam Hospital, in which we tested whether we can apply the NAB LAMP method directly on human samples. We tested nasopharynge and throat and nose swabs, and then also some saliva samples. So our first set was on the swabs that are usually taken and submerged in the media that they are submerged in. We directly took this sample and tested whether we can detect the virus there. And the next set was on saliva samples, directly on saliva samples. The throat and nose swabs are submerged in universal transfer media, UTM, and usually are taken to the standard lab Rtqpcr machine, and this machine provides a cycle.
So Naama goes on to explain that the detection of RNA in Rtqpcr begins with the RNA being transcribed to cDNA. cDNA amplification is then measured in multiple cycles, with each cycle producing more DNA until it reaches some detectable level of the target. The number of cycles required to reach this detection threshold is inversely proportional to the amount of the target RNA in your starting material, in this case, SARS-CoV-2 RNA. So the more virus in a sample results in fewer cycles to detection.
This is the standard method. We took the same samples and applied the NAB LAMP method on them, and compared it to the cycle number of the standard technique.
How do the viral loads within those different sample types compare? Do you find that in general viral loads are higher in one sample type than in another?
In our case, all the swabs were a combination. At that time in Israel, the samples were taken from both nose and throat to the same tube. So we did not compare nose and throat. And then we compared that to saliva.
Oh, okay, I understand. So nose and throat combined samples in universal transport media.
Correct.
And then saliva samples.
Yeah, sometimes it's even the same swab from both locations.
Oh, wow.
Yes, there was a shortage. Yeah, yuck, but my student that took those swabs, just not as part of this study, just as a volunteer, because he's a med student. He was saying the patients would say yuck, and he would say thanks, it's not the other way. So it was first throat and then nose.
How do the sensitivity of these assays, of the LAMP assay, compare to the sensitivity of an Rtqpcr assay?
So we adjusted the protocol, and in the adjusted protocol, we found that we can detect viral load at cycle 28 and lower, meaning viral load that is equivalent to cycle 28 or a higher viral load. Very few false negatives. And on the false positive end, we see that we didn't have any false positives, only one. And even that false positive was because we were very cautious. So in the LAMP method, as you know, the NAB LAMP method, the color change, it's isothermic and colorimetric, so we can see the change in color when the virus is present. So we first looked at the sample by eye and then also took a picture with a phone because we were very limited. We worked in the Corona lab, which is it's BSL2 plus, and we had to be very careful with the machines that we bring there. So we didn't have a very sophisticated camera, but I think it's cool because the idea was to see whether we can apply the lamp to the population, just with no equipment, no professional experience. So regarding this false positive sample, by eye it didn't look false positive, but when we took the picture and gave it to several people, some thought it's positive. So that's why we mentioned this is false positive. But in general, we're confident that the false positive rate is very low.
And is it very easy to identify a negative result versus a positive result by eye?
Yes, it's very easy. It's important to look at the sample at the very beginning. So especially if we use saliva, but also with the UTM. So it's important to compare time equals 0, the very beginning, and 40 minutes after the reaction has started. But in any case, when it's positive, it's clear yellow, and there is no problem to identify it.
That's great. So you mentioned that this might be something that people could perform on their own. Do you envision this assay being something that is a test that someone could do by themselves in their home?
Yes, I think so. It can be done at home. I would like to mention that Rich Roberts and Nathan Banner were very helpful throughout, really supporting us, and it was extremely helpful. Gave us tips and etc. And it was fun also to interact with them.
Yeah, they're great.
Okay, I would say it's possible to do this at home. It's important to have something that can either boil water or keep hot water. We think that a thermocup is the best, a thermos. It's important to have a thermometer. It doesn't have to be too precise. So the reaction is performed at 65 degrees, but it's okay if it's plus minus several degrees. However, we still need to perform a larger experiment and to get the regulation in order to approve that you can do this at home, and when you get a yellow result, you're for sure positive. And it's important to note that at this point, with the conditions that we perform the reaction, if you are negative, it's not 100% that you're negative. So it's more to find out if you are positive, but it's not yet to clear you if you're negative, because we could not detect viral loads that are above cycle 28. So lower viral loads that are still considered positive in the hospital, we could not detect at this point. So if you're negative, it's not sure you're negative. If you're positive, we are confident that you're positive. Of course, it's not bad to go and do the normal test as well, but it's kind of another surveillance. We see this as a surveillance test that can come in addition to the standard tests, because it's fast, it's easy, it's cheap. You can do it at home. You can do it whenever you're worried. You don't need to go and convince anyone to perform a test on you. All these advantages.
Yeah. And so in terms of putting together the sample into the test components, I assume that you're pipetting some very small quantity of the universal transport media or the saliva into the assay itself. So have we thought at all about how that might be accomplished at home?
Yes, there are loops. It really looks like a loop. At the end, it's a circle and a stick. We use it a lot in lab. So it's a stick, and at the end you have just a circle. It reminds me of the loops that kids use to blow bubbles. So
Sure.
So we have those, and companies create those in quite precise quantities, so quantities of volume of microliters. So you can perform this reaction at home with such a loop that picks up 2 microliters, 5 microliters, 1 microliter, whichever. We are still trying to understand which is better, but it's truly possible to use a loop of, let's say, 2 microliters and then perform all the steps only with such loops. Not difficult. So the reaction would be spit to a tube, leave the tube at room temperature for 15 minutes, then take this tube into a thermocup with boiled water. Even the boiled water, it doesn't have to be at 100 degrees. It doesn't have to be boiling, but it can be from just a bar like we have at home and at work. When you press the hot button, we did that as well, and it worked. The tube just it's better to have like a small floating piece in which you can stick the tube. The tube sits in this 95 degrees plus minus for 5 minutes, and then you add cold water until you get to the 65 degrees. That's the degree where the reaction is performed. Wait for 40 minutes and watch the color change. So after 40 minutes, you take the tube out and you compare this to the color you had at the very beginning. So it's not a bad idea to take a picture at the beginning and at the end, but it's also fine. If it's positive, you really can see the yellow color. So it's very convenient.
Yeah, that's really interesting and exciting to hear. I'm sure that, there's a very high demand for people to be able to do some self-monitoring at home, and this sounds like it could really enable that in a pretty simple way with items that people probably already have in their home. I certainly have a teapot and a thermos, so
Yeah.
sounds like all I'm missing is lamp reaction and the loop.
Yeah. And then we need the regulation. So that's, yeah, I cannot say that you can do it right now at home. We are starting a big experiment. At the end of this experiment, I hope we'll get an approval to let the population work with this reaction and decide whether they are positive or negative. So at this point, I cannot say try it at home and for sure if you're positive, you're positive. We need to perform a much larger experiment in order to get the approval.
That's really interesting. I look forward to that, and I hope that you receive approval for that test soon. Could you tell us a little bit about how you got involved in this project?
I had 2 friends of mine. With one of them, I performed my PhD side by side in the same lab, Shai Kaplan. And the other friend, Asaf Rotem, with whom side by side we performed our postdocs in very different labs, but all 3 of us are very good friends. So they read the paper by Zhang and Tanner from NEB, showing that LAMP can detect the nucleic acid of SARS-CoV-2. We talked over the phone. They asked me whether I would be interested to see whether this can work in lab. In my lab, in the settings of the Faculty of Medicine here at the Technion and adjacent to the hospital, to the Rambam Hospital, we are in close contact. So they thought if I'm interested, they thought it should be important to show the world if this works or not. So it took me several days to absorb this and understand whether this is a good idea. But quite quickly, I was happy with it and very enthusiastic. We also called Rich Roberts, as we know each other from another collaboration that we're performing for several years now on a very different topic. And after talking with Shai and the staff, talking with Rich, thinking, and I think quite quickly all the lab was very enthusiastic about it. So quite a few students and myself, my lab manager as well, came to lab and almost didn't go home for about 2 weeks and very, very few hours of sleep. And I would say that the Rambam Hospital was crucial here as well, because very fast they were also very enthusiastic and did everything they could to enable this experiment, these tests. Also, the Technion, the response, we had to get approval because it was during the lockdown phase of the labs. So every experiment or every project had to get approval. So the response I got was, wow. It wasn't the word crazy, but it was, this sounds wild, but wild make a big leaps in science. So just go for it. And that's how it started. And with all this enthusiasm, we worked really very hard, but also very happy to do this type of work, because we really felt it can help the world. And after 2 weeks, we got this paper written with over 200 different samples. I have to say the saliva test was also quite an adventure, because we got approval for a few saliva tests. We had to find someone that will agree to give us his or her saliva test, because it wasn't the standard. So it was also kind of a mission. I put Inspector Gadget on our group icon.
Yeah, it sounds like a very exciting time for your lab. And we really appreciate all the hard work that you and your team have been putting into developing new diagnostics, new tests for COVID, to really help the world recover from this pandemic. So thank you so much for your efforts.
Thank you, and thanks Annie B for this lamp reaction. It's really it has a lot of potential. I can also elaborate that we also tested different viruses with this method, and the method only picked the SARS-CoV-2, even other RNA viruses, DNA viruses, and we didn't get a positive result on them.
Awesome. I know our scientists are really proud of it and happy to have all these collaborations and to see all these different applications of the method, hopefully help the world move on from this pandemic, move forward in a safe and healthy way. Well, thank you so much for joining me today, Naama. It's been really interesting to hear about your application of the LAMP assay. And I look forward to hearing more about it in the future, and hopefully hearing about an authorization for diagnostic use.
Yes, we're waiting for the authorization, but I'm optimistic.
Excellent. Well, good luck.
Thank you.
Thanks for joining us for this episode of the COVID-19 Researcher Spotlight Series. At the time of this podcast release, New England Biolabs currently has made available a research-use-only SARS-CoV-2 rapid colorimetric LAMP assay kit, and is focused on supplying research-use-only reagents to companies developing diagnostics and vaccines. Join us next time when I interview Dr. Josh Quick, a postdoc at the University of Birmingham in the UK. Josh's work focuses on using genomics to inform diagnosis and surveillance of infectious diseases. His experience began with the West African Ebola outbreak in 2015 and continued with the Zika outbreak in Brazil the following year. During the current COVID-19 pandemic, Josh has been an important part of the Arctic network, which early on made available information like primer sets, protocols, and data sets to assist researchers in sequencing this virus.